Hello, I'm Tom Neer, Professor of Ecology and Evolutionary Biology, Head of Sabre College, and the Bingham Oceanographic Curator of Ichthyology here at the Peabody Museum. The snail darter is a freshwater fish species that was discovered in the Little Tennessee River system in the mid-1970s. The snail darter is really important in the political life of endangered species, is that it was the first test of the Endangered Species Act that was passed and signed into law in 1973. The only known habitat of the snail darter at the time was endangered by the construction of a dam that was going to create a reservoir, an artificial lake, that was to obliterate all of the known habitat of the snail darter, which was listed as an endangered species and protected. The case went all the way to the Supreme Court, and the small fish, which measures about 3 inches in length, actually won in this David and Goliath contest between the small fish and the very powerful Tennessee Valley Authority. However, the dam was ultimately built because Congress put a rider on a spending bill that was signed into law by President Jimmy Carter, exempting that specific dam project from the Endangered Species Act. Our research showed that the snail darter is not morphologically or genetically distinct from a species described in the 1870s, the stargazing darter. So what that means is that the distinctiveness in the snail darter, the strong conservation protection that was aimed at the snail darter, may have been misapplied. So the method of species discovery and delimitation that we outline in our study of the snail darter can be applied across any range of plant or animal species. And more often than not, it results in the discovery of new and unnamed species that require further study and names so that they could be in the catalog of biodiversity, but also can result in the collapsing of species that are not distinct from each other. And this is important because this will inform our application of the limited set of resources that we have for the conservation and protection of endangered species. There's an excitement in scientific discovery. And that excitement comes when we gain knowledge that essentially upends what we previously thought. So in the case of a biodiversity scientist, particularly one who studies freshwater fishes, we have range maps for any number of sets of species. We may see a species of freshwater fish that's widely spread across eastern North America, the rivers and streams of eastern North America, and through the application of DNA-based evidence, reassessment of traditional morphology, we can see that that species is actually 5 species. And so that result, we've discovered 4 new species to the biodiversity catalog. Often local stakeholders are excited when you can tell them that there's a species found nowhere else in the world, but in their part of the planet. So whether it's a species very closely related to what may be among the world's most popular freshwater sport fishes, the largemouth bass, there's a bass species that's found nowhere on the planet but the hill country of Texas. And there's no one more proud of this species than the folks who live in Texas. Or when we travel to the Eastern Highlands, the Appalachians and Cumberland Plateau, and tell local residents who we interact with in our field work, we show them these species of fish that are found nowhere else in the world, but maybe in a small 50 square kilometer drainage area. Invariably, you could see people's pride elevate. People are excited to know that where they live, what they think is special, has also some special aspects of biodiversity. My lab's work here at Yale is seen in every room, every corner of the Yale Peabody Museum. But also the activity of scientists here at the Peabody utilizing our deep collections, which essentially are libraries of biodiversity, is resulting in dramatic discovery of biodiversity, living biodiversity, here in the 21st century. As we intersect the traditions of scholarship and morphological traits that we could use to study living things as well as the extinct things like our dinosaurs, we're intersecting that with genomics to where we have a 21st century approach to discover and delimit biodiversity, making the Peabody a hub for that activity here at Yale and New England broadly.